Good evening and welcome to the sixth week of Happy Feet. Today we have our speaker is Dr. Kelly Lambert. She is the Tralwick Professor of Behavioral Neuroscience at the University of Richmond, where she investigates experience-based neuroplasticity and teaches multiple neuroscience courses. She has approximately 80 scholarly publications and has written two neuroscience textbooks and three mainstream books, including Lifting Depression and The Lab Rat Chronicles and Well Grounded. She is currently writing Wild Brains, translating adaptive mental health strategies from the bush to the bedside. Dr. Lambert was awarded the 2008 Virginia Professor of the Year Award and in 2018 Career Achievement Award given by the International Behavioral Neuroscience Society and the 2024 Science Educator Award by the Society for Neuroscience. During the 2024-25, that's a lot of 20s, academic year, she visited campuses across the USA as a Phi Beta Kappa visiting scholar. Her research targeted connections between physical effort and emotional resilience and has been featured on CBS Sunday Morning, and her rat driving research has been featured on 1,500 news stories across the world, including the Netflix series The Hidden Lives of Pets and the Canadian Broadcasting Corporation's Rat City. In 2019, Dr. Lambert described her behavioral research perspective in a TEDx Bermuda talk. And that's where I found you, Dr. Lambert. And I think you said you coined the word behavioral, right? So we are excited to learn more from you, and I'm going to unpin myself.
Thank you, everyone, for being here on this starting to be a rainy, sunny evening. It's rainy Sunday evening here in Richmond, Virginia, as well. Heard that it's raining for you, and I'm a little bit of a different researcher than maybe you've had or speaker, in that I'm a behavioral neuroscientist. So a lot of the wisdom that I'll be sharing comes from unexpected critters, rats. They're my colleagues. And if we're finding things in rats, it suggests that they're universal findings. And then maybe, well, I've learned a lot from rats throughout my life. So feel free to stop me at any point. And we certainly will have time for questions as we go through. But just to describe what I do here at the University of Richmond, teaching students and doing research, behavioral neuroscience really considers these three different pillars: the brain, environment, and behavior. So I'm very interested in how the environment, the context of our lives, the behavior that we engage in, how that influences the brain and how the brain influences our behavior. So we use animal models to try to understand more of that, but certainly there's research done with humans, but we can't do it to the same extent as with our animal models. And a lot of my colleagues, and we're gonna do a little bit of a switcheroo on the topics as we get to the end, but most of my colleagues and a lot of my career has been spent studying the more negative emotions, the sad, the anxiety, not so much the anger for my lab, but fear. I guess this is fear. I forget their names here. Anxiety was added later, and it's for good reason, because some of those negative emotions start to interfere with our functioning. And even though there's been such a lot of research in depression, and depression is something that my students and I are interested in, uh, it's just so odd that we just are not making progress in having or developing effective interventions. We have medication, and that works for some individuals, but there's still a lot that we don't know. And so we see, especially after the pandemic, we saw a jump in depression rates. And we don't see these for other medical conditions, cardiovascular disease and such. We see improvements and less incidence rates. So it's a big mystery with psychiatric illness. While we're not doing better, we need to do better. Um, and so with our models, there are a lot of aspects to depression. We all have had low mood. Maybe it's not and hasn't been diagnosed as depression, but we all can relate to having those low moods. But some of these symptoms that are associated, that we're familiar with, metabolic changes, changes in appetite, sleep disturbance, agitation, fatigue, difficulty concentrating, and then I would add on some anxiety, and that anxiety is something that we've been interested in. One of my favorite researchers, Dr. Robert Sapolsky from Stanford University, has a great podcast as well. He describes depression as an inability to appreciate a sunset, and I can relate to that as well. It's hard to test in the rats that. And Peter Sterling and Michael Platt a few years ago published a study in the Journal of American Medical Association, a very sad study, and we are going to get a little bit more positive as we go through. But looking at what they call deaths of despair, deaths by suicide or related to addiction or metabolic syndromes that seem to be anxiety related, those deaths are increasing. So these more negative emotions and conditions certainly deserve our attention. Um, and so we've looked at, what could we understand about depression in a rat? I'm not saying that a rat gets depressed, but we can start to look at some of the symptoms, such as anxiety, maybe lack of activity or motivation, lack of reward, and that would be another symptom that I would put up there, not going along with that sunset. And with the animal models and with fMRIs with humans, we do know that depression and low mood is the brain is kind of all in. A lot of different brain areas are involved. Our prefrontal cortex that helps us with the cognitive decision making, part the what we call the limbic or emotional kind of module, involved in fear and in memory and motivation. So there are a lot of areas that we can look and that we have looked with the animals, um, to see, what is involved in this, I'm going to say sadness and lack of reward and anxiety, a very complex situation and condition. So if we know what brain areas are involved, where does that leave us? We can't really go in and infuse more brain power there or cut something out that may be giving us some trouble, for a condition such as depression. So we have to go to other types of interventions. And the toolbox that has been readily available and is available is the pharmaceutical toolbox. So since the 1950s, when we gladly stepped away from frontal lobotomies, which was quite invasive and not a great intervention, the idea that you could take a pill and change the neurochemistry, we've all heard that there are imbalances related to depression and that we could reestablish some of those imbalances. It just, that would be great if we could do that. However, I've been frustrated with, there aren't clear indications in the literature that everyone with depression has that chemical imbalance. It's kind of a indirect effect. So whereas it may help some individuals, it hasn't been the magic pill, the magic bullet to help everyone. And sometimes there are side effects that can be problematic as well. And I'd like to offer the disclosure that I am not a clinician and I'm not a psychiatrist. So I'm just sharing information that I've learned through the literature and through our research. But any decisions about changing any type of medication related to mood should be done in accordance with your prescribing physician or your therapist. So turning to the animal models, we wanted to see if we could look at some interventions, and then have some cases where the animals could show us that they were feeling better or they were more resilient. And emotional resilience is kind of part of chipping away at this depression so that we have this confidence. So these are my colleagues, the Long Evans rats. So they don't look like the rats going around downtown Richmond here. And we are trapping wild rats to compare them, but that's another talk for another night. These are our very smart, selectively bred Long Evans rats that we work with. So as I started to think about a different perspective for depression. I'd written a clinical neuroscience textbook, and when I wrote the chapter on depression, it was not encouraging. I was like, there's so much research. Why, we need to do better. So I tried to take a different perspective, go to the brain chalkboard, and I became interested in the idea of movement and how much of our brain is related to movement. This cerebellum hanging off the back of the brain, almost like an afterthought, it has 78% of all of the brain cells or neurons, is what we call them, in our brain. So we like to think of that outer cortical area like bark on a tree having more of it because it's related to more executive functions. But this cerebellum has more neurons, and it's involved in motor coordination, balance, also some functions related to conscious awareness and basic learning, but movement. So that's a big chunk of our brain related to movement. And then in the middle of the brain, the striatum here, again, taking up a lot of territory and real estate, is involved in smoothing out and initiating movement. So individuals with Parkinson's disease or Huntington's disease, this area of the brain is affected. And then there's the motor cortex kind of going here, and it's involved in voluntary movement when you're telling your arm to move like I am, or hands, or when you're walking and initiating movement. That motor cortex is sending messages to the appropriate muscles. So movement is incredibly important for the brain. And when I started to think about movement, and I was thinking, what has changed if depression rates are increasing? What I don't think we've had a genetic mutation or something like that that's happened in the last 50 or 100 years, but something that has changed is our movement. And this is from the New York Times, as you see here, in 1939, when the World's Fair introduced the television for the first time. And they wrote in the New York Times, the problem with television is that people must sit and keep their eyes glued on a screen. The average American family hasn't time for it. For this reason, if for no other, television will never be a serious competitor to radio because people are just too busy to sit in front of a screen. But boy, we showed that they're wrong. We have multiple screens. I'm looking at three right now, and we're just happy to plant our rear ends in a chair and watch these screens. So interesting. So my point is that our movement has certainly changed over the last century as we become more technologically advanced. I don't know if our brain has interpreted it that way. Also, the way our motor cortex is mapped out, it has a certain amount of neurons that control different body parts. And so a large part, a proportion of these neurons go to our face and our hands. So tasks or any activities that involve our hands are activating a good portion of that motor cortex. So hobbies and such could really activate the brain in interesting ways. This set me off to look at different types of hobbies or activities that involve the hands. Also, the hands, it's the right hemisphere is controlling the left. It's what we call contralateral. So you're engaging with both hands, both hemispheres. And about 100 years ago, there was a book on the history of knitting. It took me a lot of coffee to get through that book, but it was a real gem in there talking about how doctors would prescribe knitting. At that time, they said to women who were um, uh, wrought with anxiety, as they said, they sensed that the knitting calmed them in some way. They didn't dissect it or pick it apart, but just kind of threw that out there as some type of intervention. And so if I can't get my rats to knit, I'm going down this path to show that we're going to get them moving in some interesting ways. But I can think of how knitting using both hands. Also, you concentrate on your stitches so you're not thinking about what may be worrying you at the time. Also, as we go through this, I'll talk about social benefits, and oxytocin is a neurochemical that can reduce stress hormones, and stress hormones are a big part of depression and certainly anxiety. So the rhythm of the movement. And then if you're knitting something you like, some of it can be rewarding. So I can dissect that now, what the doctors were saying 100 years ago, based on what we know about neuroscience, and say, yeah, that makes sense. And in my book, Lifting Depression, I shared a story. And so I started to think about these, just a lived experience. And I don't enjoy vacuuming, but this story is about vacuuming and how it helped me when my mother passed away when I was 32 years old, and just, yeah, like most people would be, in a very low mood, maybe depression. I wasn't diagnosed. And at that point, going back and lecturing about neurons and ions and all of that didn't seem very relevant or rewarding to me. But what I wanted to do was just something where I could see the impact. And I just heard about these rainbow vacuum cleaners. I don't know if anyone's heard of those. And it was something that we couldn't afford on an academic professor's salary. Um, but it has this, I don't know if you see my pointer, but this isn't a rainbow. But the idea is it has a tank with water in it, and it sucks up the dust so that it's not like all in the air. And you could dump this pile of dust in the backyard, and you can see, oh, my effort, I see an effect. I'm cleaning the house. I'm getting germs out of my family's house. I'm doing something that seems to be productive. And so after my mother died, we were in at my husband's family in Georgia, and there was a big flea market, and we got a repurposed rainbow vacuum cleaner for a very cheap price. And I started vacuuming behind the washing machine, behind the couch, and the pile grew bigger and bigger, and it seemed to really help me take baby steps toward a world where I felt like I had a little control. This is just one example. It may be something else. And my husband does the vacuuming now. I don't like it, but it got me through a hard time. And I realized that physical effort and interacting with the world there seemed to be something to it. So I wanted to test drive this with the rodents. And we came up, I call it effort-based rewards. Our rats love Fruit Loops. They're the currency of our lab. And so we teach them, first we get them a little, well, where they like, I was going to say addicted, that doesn't sound great, but they will work for Fruit Loops. And then we put them in an arena that has mounds of bedding, and we gradually bury the Fruit Loops so they know when they see one of these mounds, they go and just gently dig, and they're going to get that reward. And so over a week or so, we teach them, and they will reliably go, and they have 4 opportunities to get a little piece of a Fruit Loop. They get a nutritionally balanced diet as well. So we had to do a proper experiments. We had the experimental group, and these animals only got their Fruit Loops if they went and they dug them up. So they had to work for them. And then the other group were just given the same number of Fruit Loops. So it's contingent and non-contingent. So these guys, they got their Fruit Loops regardless of what they did. Kind of looks a little guilty there. Like, I didn't earn this. So with students and I, we enjoy calling the ones that work for the rats, work for the Fruit Loops, the worker rats, and those that just have them handed to them, the trust fund rats. So we put them to a test, some different challenges, like in an aquarium. They're very good swimmers, but they don't love swimming. So looking at if they would swim or float or dive, if they would um, change their behavior. We look at stress hormones. We gave them some spatial tasks to see how that memory was going. We looked at their stress hormones. So we're getting at little bits and pieces of symptoms of depression without saying that a rat is depressed. Um, so we saw increases in biomarkers of emotional resilience, such as a higher proportion of a healthy stress hormone called DHEA that is released from our adrenal glands in accordance with cortisol, which you have probably heard of, which is our more stereotypical stress hormone. Stress is good and low mood is good. They all play their role, but when the tipping scale gets a little, if it overshoots itself, that's where some problems can step in. So we saw healthier profiles in our worker or contingent rats. They were building sense of competency as they understood their world and how they could interact with it to get their rewards. So a little bit of confidence and certainty about their environment. And they also did something that just blew my mind, and an artist captured it here. They would dive down into the water, which is very confident. They'd never been in water before, and they don't know, they hold their little nose, but they just go down and look for an escape route. And that's just so bold. And we're looking for bold responses in these animals as they build confidence with the world around them. So training and working. So this is one of the another biomarker in addition to the stress hormones is a growth factor, kind of like fertilizer in the brain. This is BD, brain-derived neurotrophic factor, or BDNF, and um, so we saw more cells that were producing that. And if we look at what antidepressants are doing, if we give a rat an antidepressant, it's like is their book, Give a Mouse a Cookie or something like that, it will increase some evidence of markers of neuroplasticity or the brain growing in some ways. It takes 3 to 4 weeks or so in the animals. It doesn't happen automatically, and that's probably why antidepressants don't work automatically. The more traditional antidepressants, other psychedelics like ketamine may change things more quickly, but we're still trying to understand what they're doing and the effectiveness for long term. So we're seeing some of that in just good old-fashioned behavioral training and activity, seeing some of the same effects as some of the drugs. Also, we gave them some tasks to see if they would stick to a task longer, as if maybe they believed in their efficacy or as anything they would do and make a difference. So this is not very high tech, but sometimes more simple things are better. We just went to the pet store to get a little cat ball, and it has a Fruit Loop in it, but it's too big to come out of it. So we had opened it up to get it in there. So they bite it, they try to throw it against the wall and such. We were interested in how long do they keep trying to get this? Because another symptom of depression is kind of giving up and not making that effort, not getting out of bed to go for a job interview or interact with friends or such. And our worker rats, the contingent effort-based reward trained rats, they stuck at this task longer, as if they had a belief that their interactions with that object would result in a reward. So behaviorally and neurochemically, neuroanatomically to some extent, we were seeing some big differences just with behavior. Um, and this was more of the stress hormones that I talked about. And yeah, so the contingent, this DHEA to court ratio, I know it's Sunday night and we're tired and this is a little too much detail, but the higher the ratio, the healthier. And so we did different baseline and then a first swim and then a second swim. And the contingent rats kept getting better on that healthier profile. The non-contingent, the trust fund rats, they just stayed the same. So interacting effectively seemed to make a difference. So this is a rat floating, and floating is a very adaptive behavior too, once they discover that they can't get out. And we only leave them in there for 3 to 5 minutes, and then we take them out and warm them with a nice towel. We were always interested in if this generalizes to humans. And there's been some research with special operations soldiers where they do all this training and throw them in the water, sometimes with their hands tied behind their back and things that we wouldn't even be able to do with rats. But they really challenging them. And they see the healthier stress hormone profiles in those trained soldiers as well, but also I consider the diving to be a flexible coping strategy. And that seems to be really important. When we get kind of in a rut and we keep doing the same things over and over, even though they don't work, um, that's because our higher levels of our brain aren't navigating the system very well and in control. So being able to leave a pattern of behavior that isn't working well and trying something new is really important. And the animals that had more confidence and were trained did better, showed us more flexibility with several different tasks. So that seems to be important. And as Jen mentioned, to try to make it sound as important as drugs, I just made up the word behaviorseuticals. And the idea, this was one artist did this, that maybe it's not the pills in our cabinets, but the crafts and other things that can intentionally change our neurochemistry, our neurophysiology, even growing new neurons or making new connections. Um, I just think that's some powerful stuff. Um, a challenge is you're not going to make the same amount of money as the pharmaceutical companies are. It was interesting when this started to get out, some of the articles and some real world responses to it. Carol here, I read in Woman's World, which isn't my typical science journal, but I loved seeing this. Um, she went through a really tough time losing her daughter. I think I remember, I'm not reading all this now, and she started quilting and said that that kind of brought her back to, recalibrate it a little bit. I know still very sad, but she had read Well Grounded, my recent book about this, and so many individual differences. So maybe knitting, maybe something different. This is woodworking. Um, and Patrick Wells, he contacted me from London, and he had been a war correspondent and very, very stressful. And when he got back to London, he said he hadn't been diagnosed, but he thought that he probably had post-traumatic stress disorder. And he had read about effort-based rewards and decided that instead of doing the traditional, maybe pharmaceutical routes, which should be done in accordance with a physician, he decided to build a boat by hand. And he reported that that just made him feel so much better and more in control, to the extent that he stopped being a war correspondent and he started a new business called My Own Two Hands. My Own Two Hands. And he uses his film expertise to produce videos about how to do projects. So he has like kits for making baskets or woodworking, and he sends the tools and he sends the video. And so individuals can have that making experience that he thought was good for his brain. So I love it's rewarding to work with rats, but it's super rewarding to see that this has some influence on humans. Uh, and this was years ago in Scientific American. I wrote about this, and their authors they called it, used the title "Depressingly Easy." And it's interesting to think that as we think our world has become more advanced, where we just press a button to get everything, even for our social interactions. I mean, this is good that we're connected here, but this young woman in a kitchen where it's just so sterile and everything is probably, could be delivered and you don't have to do so much. Maybe it's that work, all of that stuff that we've tried to be so clever to not have to do, that our brain is still craving. We're still walking around with the brains of our ancestors. So it may take them, I don't know if they'll ever come up to speed, not having to engage um, in life the way our ancestors did. So yeah, that already showed that. Also, with our effort-based rewards, I mentioned, ketamine. And we're not assessing that right now, but I was interested in what ketamine may be doing to see if our behavioraceuticals could be addressing that. And there's this little bitty brain structure called the lateral habinula, and it is active when we are very frightened, kind of have a high anxiety, kind of deer in the headlights kind of response, a freeze response is what we call it with the animals. You just can't move. It's adaptive to not move when you're a bunny rabbit and you don't want the predator to find you, but it's the brain's area that does that. So it is relieving, diminishing some of the activity ketamine does in the lateral habinula. And we saw some effects in the lateral habinula of our rats, where there was less of that neural growth factor, the fertilizer, so that area of the brain was not as active. It's meeting a lot of the criteria that we're seeing in some of the other approaches. So generally, just doing a task, an activity where you feel like you're gaining a sense of control. It may even be coloring. It could be baking. It could be a lot of different things. Increased persistence in these animals, increased cognitive flexibility, increased markers of neuroplasticity, and less reactive, this brain area involved in kind of this stop in your tracks, overcome with anxiety. So I think there's a lot of promise to behavioral approaches to mental well-being. I have called this the prehistoric Prozac. This is probably the antidepressant that our ancestors used. Um, and so we're doing some other things in the lab, looking more at this experience-based neuroplasticity, literally sculpting the brain with our experiences. It's never too late. I always tell my students, our brains are changing from the womb to the tomb. So if we're alive, our brains are changing. It could change both ways, maybe not so great and in great ways, but it does change. So another way we've looked at this is to put rats in just Disneyland kinds of environments so that they're more engaged. So it's similar to what we saw with the training, effort-based reward training, but it's less systematic. They're just exploring things on their own accord, so to speak, to see how pushing something results in something. They're also more active. They have their friends with them, so social interactions. And there's been a long history of research, we call these enriched environments, and it enhances neuroplasticity. So we know that that has been working. And just to kind of give you, I have a lot of respect for animals, and we try to give them the best life we can in the lab. And I know that can be an kind of controversial thing to use animals in research. But I like to use some form of enriched environments for all of our animals. But this is the way most lab rats are housed. There is food and water, but not in a very big space. So even in our control conditions, we try to have larger environments. But something different that we've done, this is kind of the more manufactured enriched environment. We've looked at natural enriched environments to compare to that. So we have dirt that they can dig in and wood and sticks instead of plastic, and we control for the number of items. Uh, but we have found, and others have found, that just being in a complex environment where there's something going on, there's something interesting, that you're learning something, increases these bright cells are new cells, neurogenesis. In this particular example, they looked at ones that had, or rats that had a running wheel versus not. And so with the animals, the number one way to increase neurons is running. So get those sneakers on. I'm not sure we actually have, people say you have to run, but walk fast, just move. That results in more brain cells as well. And then I know that this gets complicated too, but this is a wonderful, when I read thousands of articles to write my this textbook, this was the article that stood out the most that Kent Berridge did. And he exposed animals to not even enriched, but a natural environment that didn't have the harsh lighting. They had another environment that was stressful. It had music and louder and brighter lights. But in the more natural environment, he was able to look at this brain structure that's involved in reward. And with depression, we seem to have a lack of reward. And the cells, if you kind of spread out that structure, the cells that would respond to a stimulus in a positive way were much more prevalent than in the stressful condition. So green is positive responding, red is not. So this shows you that the world you live in can actually sculpt your brain so that it is more or less likely to respond in a positive way to a stimulus to this area. That just blows my mind. And he used the terminology about the environment and our behavior tuning, fine tuning our brains like a piano could be tuned. And I just love that. And also in the enriched environments, as I mentioned, they have their rat friends. And we've spent a lot of time looking at parenting and social impact. And again, I won't go into that so much, but the brain changes when we are engaged in social interactions. It doesn't have to be parental, and it doesn't even have to be the same species, apparently. Our pets, I see a kitty cat there that I'm loving seeing. That is increasing. I see you petting too. Oxytocin that I mentioned a while ago for both you and the cat, probably. And that will reduce some of that cortisol and it's also rewarding. So social interaction is so important. And as I mentioned, the exercise that goes with the enriched environment is going to kick up those new brain cells that are going to help you problem solve in times of low mood when that outer executive function may not be as activated. So these are all called plasticity-based therapeutics. They're not drug, they're not surgery. They're moving about and interacting with the environment. I wanted to leave you with getting to the joy, a more positive note, which we've been going a little positive here. Something that happened during the pandemic that really changed the our research. And I want to do it backtrack a little bit. Jen mentioned our driving rats, and yes, we had I'm here with undergraduates, and I was a new professor here after a long career somewhere else. And a colleague asked me if I could teach a rat to drive a car, and I thought that was absolutely ridiculous. I was much too serious-minded to do that. But then once you start thinking about that, you can't stop thinking about teaching a rat to drive a car. But we started really simple with a cereal container from Walmart, and she did some robotics, and we actually had them steering again to get to the Fruit Loop tree. And we also found that the animals in the enriched environment learned faster than those in the control environment. So we were doing that, and as Jen said, it's become great outreach. But there was something happened one morning when I came in. We were taking turns during the pandemic when the students weren't here, and it was my turn to go in and feed the rats. And this helped to shift us from that sad to joy. And I walked in, and the rats were on the cage like this, like they were excited to see me. And I was so, the way we were in the pandemic, I thought it just made me so happy that they were excited to see me. It made me feel good, and they were reaching out and jumping up and down. Now they probably just saw me as a big Fruit Loop or something, but it looked like they were happy, that they were excited. This word joy, could I talk about joy with a rat? Can we define that? But it certainly looked like when I would ask my dog Brody if he wanted to go for a walk, and he goes around in circles and he yips and he jumps up and down. That's what these rats were doing. And so much of our field is about, studying those negative emotions and inside out, stress and trauma and fear and anxiety. I just decided then I'd had enough, and I wanted to, whatever this was, I wanted to study it. So we came up with protocols where we expose our rats to three fun things every day in an unpredictable pattern. So they don't know when it's coming. So what we're trying to do is stretch out positive anticipation. Call this unpredictable positive emotion responses. So my behavior pseudocol upper. Um, and so we would give them a Lego block, and then they would have to wait 15 minutes to get a Fruit Loop. We would give them sunflower seeds that were shelled, so they'd have to wait to kind of undo it to get the seed. And then the most fun, they would go to Rat Park. We put them in a transport cage, and then they went to an enriched environment where they could play. They had to wait in the transport cage for about 5 minutes, and then they would get to go in this park and play for 30 minutes or an hour, depending on the study. And so from the beginning, the upper rats, when we walk in, they would do this on the cage, like they were more excited than the other animals that were not on the cage as much. But we were studying other areas of the brain. There's a rat version of optimism, you believe it or not, that you can study something like that in a rat where they have an ambiguous cue, like a grid, after they've been trained with horizontal and vertical lines to mean certain things. So this is an ambiguous cue, and if they interpret it with a positive outcome, that's optimism. Now, I won't go into all the details, but that's the way we would do something like that with a rat. So just to show you what this anticipation is, these are three rats waiting to go to the park. They are so excited. We're not giving them cocaine. They're not getting fed. They are just so excited. Always, look like Swifties standing in line for the Eras concert or something. So this is doing something to their behavior. The mere anticipation. Also, their tails started. The student said, "Dr. Lambert, why are the rats' tails up like an umbrella handle?" Oh my gosh, I've been studying rats for 3 decades. I've never seen that. But I asked some of my colleagues, and they said, sure enough, this is called Straub tail. And if you inject mice with opiate drugs and or dopamine that's involved in reward, their tails will do this. We didn't inject our rats with anything except positive experiences, and they were showing something that's similar to a pharmaceutical intervention. And one of my favorite human studies, I mentioned my favorite rat study with the fine tuning the brain. An Israeli study was published looking at outcomes of children who had been diagnosed with cancer, who had been granted their wish, and then they waited for about 5 months before between knowing what their wish was and then going to Disneyland or meeting the celebrity or athlete. And they were measuring their mental well-being and some other outcomes related to health. And they compared them to the children who had not been grant. They didn't know what their wish was. They were still in the queue. They knew something may be coming, but it wasn't something that they could really, expect in the way that they could with the others. And um, their outcomes were significantly different. The ones who had been given the wish and they were looking forward to it. This anticipation is changing some things about the brain, stress responses that can have some overall health benefits. And I'll leave with this. A lot of people ask me, Kelly, do the rats like driving? I can't give them a survey or anything, but what we did was we gave them the choice of walking to the Fruit Loop tree, which was an easier path, or backtracking and driving. And let's see if this will um, so the majority of the rats, just a pilot study, they didn't walk, they ran. They're a little chubby, and drove to the Fruit Loop tree, even though they could get to it faster. Whoops, this is something has changed here. Let me, but that was I'm just going to go to my slide. Oh, and now it's doing this, but I will, that's okay here. So I think the takeaways are what the whisker wisdom is telling us is that movement is important, the natural enriched environments, and it may be like I've got sitting here in my office with plants or taking a walk in nature. And I should have mentioned that the animals in the natural enriched environments had more emotional regulation, social connection, purposeful activities, and planning something that you're looking forward to. That's what the rats are telling us is important. So I appreciate the contributions of the rats and my undergraduate students who have been a great help and support from the University of Richmond. And I appreciate your attention, and I'd love to answer any questions that you may have.